Hello students, in today's class we are going to learn about restriction enzymes and enzyme DNA ligase. So restriction enzymes are enzymes that cut DNA at specific nucleotide sequence which are known as restriction sites or recognition sites. They are found in bacteria and have evolved to provide defense against invading viruses. In bacteria, they selectively cut up foreign DNA in a process which is known as restriction. In order to cut the DNA, restriction enzymes make two incisions, one on each strand of the DNA double helix. This slide represents how the restriction system is functional in bacteria and is used by them for protection of the self DNA and prevent infection against invading viruses. There are specific sequences that are present within the bacterial genome which read as GATC regions. The adenine residues of these sequences are methylated. So here we have parental DNA strands in which both the strands of DNA are in a completely methylated form. During replication, we have synthesis of a daughter DNA using the parental DNA as a template. So for some time initially, we will have the DNA in a hemi-methylated form where one strand is methylated while the daughter strand is unmethylated. Later on, towards the end of the DNA replication, both the strands become completely methylated by the activity of enzyme DAM methylase, which makes use of S-adenosine methionine as a methyl group donor. So at this stage, we are unable to distinguish which is the parental strand and which is the daughter DNA strand. Hemi-methylated and completely methylated forms of DNA are considered as self-DNA, while unmethylated forms of DNA are considered as foreign DNA molecule, and they are restricted by the bacterial restriction enzymes. And in this way, the bacteria protect themselves against invading virus particles or other pathogens. The sequences that are identified by the restriction endonucleases are basically palindromes. So here we have normal palindromes that are formed by alphabets, and as you can see, they read the same as we read it from forward as well as in the backward direction. Such sequences are known as palindromic sequences. So far, about 3,000 different restriction endonucleases have been identified, and about 600 of them are commercially available and are routinely used for restriction systems or for genetic modifications. Restriction endonucleases can scan the DNA sequences and cut the DNA at specific set of nucleotides by making specific cuts. So here is an example of a genetic palindrome, and as you can see, the sequence reads as GAATTC in 5' to 3' direction, and it reads the same on the other strand in 5' to 3' direction again. So they are also known as genetic palindromes. So restriction enzymes basically recognize and make a cut within the specific palindromic sequences, which are known as restriction sites, and they are generally 4 or 6 base pairs in length. There are 3 different types of restriction endonucleases: type 1, type 2, and type 3 restriction endonucleases. Type 1 restriction endonucleases generally cleave at a random position about 1000 base pairs away from the recognition sequence, while type 3 restriction endonucleases cleave about 25 base pair away from the recognition sequence. Hence, type 2 restriction endonucleases, which recognize and cleave within the same sequence, are most applicable with reference to recombinant DNA technology. So this slide shows us the comparative properties of the 3 types of restriction endonucleases: type 1, type 2, and type 3 restriction enzymes. And type 2 restriction enzymes are the most common type of restriction enzymes. The recognition site identified by restriction enzyme type 1, it cuts both strands at a non-specific site, which is located about 1000 base pairs away from the recognition sequence, as we just discussed. And type 2 will digest within the recognition sequence, while type 3 will digest 24 to 26 base pair downstream of the recognition system, of the recognition sequence. In case of type 1 restriction endonuclease, we have a single multifunctional enzyme that is able to carry out both restriction as well as modification function. We have two separate enzymes in case of type 2 restriction enzymes for the two functions, restriction and modification. And we have again two separate enzymes sharing a common subunit in case of type 3 restriction enzyme. The nucleus subunit in case of type 1 enzymes is a heterodimer. It is a homodimer in case of type 2 enzyme and a heterodimer in case of type 3 enzyme. Type 1 enzymes require ATP, Mg2 plus ion, and S-adenosylmethionine as a methyl group donor as cofactors for their activity. Type 2 enzymes require Mg2 plus ions, while type 3 enzymes require both Mg2 plus ions and S-adenosylmethionine for their activity. The DNA cleavage requirements in case of type 1 enzymes include two recognition sites in any orientation. Type 2 recognition restriction enzymes require a single recognition site, while type 3 restriction enzymes require two recognition sites in a head-to-head orientation. DNA translocation is observed in case of type 1 enzymes since the site of recognition and digestion are quite at a distance from each other, while the type 2 and type 3 restriction endonucleases do not show any DNA translocation reaction. The site of methylation is at the recognition site in case of all three restriction enzymes. So the restriction enzymes basically recognize and cut the DNA within the palindromic sequence, which is known as the restriction site, which is usually 6 to 4 base pairs in length. So let us look at a few examples. So here we have an example of H3. So H3 will recognize a 4 base pair sequence. The sequence that is identifies is GGCC read in 5' to 3' direction. And it cleaves at the axis of symmetry. So these are the ends that will be generated after restriction digestion. These type of ends which are not having any bases hanging out without base pairing are known as blunt ends. Then coming to the nomenclature of the restriction enzymes. So the nomenclature of the restriction enzymes, first of all, also depends on the source or the name of the bacteria from which it is isolated. So ECO stands for Escherichia coli. R stands for the strain of E. coli from which it is isolated, and 1 stands for the first enzyme that has been identified, first restriction enzyme that has been identified from E. coli. Here are some examples of restriction enzymes that will generate sticky ends. So H3 we are already looked at, which produce blunt ends. And here we have EcoR1 restriction enzyme recognition site, which reads as GAATTC in 5' to 3' direction. The digestion is between the first two bases, G and A. And the ends that are generated will be having few base pairs, few bases that are not pairing, or they are basically hanging from the ends of the digested DNA fragments. So these type of ends that are generated are known as sticky ends or cohesive ends. And these enzymes are known as sticky end cutters. This slide just shows a diagrammatic representation of how the blunt ends are and how the sticky ends are. Here we have few more examples of restriction endonucleases, where you can see that HND3 and BAMH1 are enzymes that will generate sticky ends, while ALU1 is an enzyme that will generate blunt ends because it digests at the axis of symmetry. Two more terms which are associated with restriction endonucleases include isoschizomers and neoschizomers. So isoschizomers, so restriction enzymes that are from two different sources. For example, SPH1 and BDU1, and they recognize the same restriction enzyme target site and also digest the recognition site in the same fashion, generating same type of ends, are known as isoschizomers of each other. Similarly, there are some restriction enzymes that are again from different sources. For example, SMAV1 identifies CCCGGG sequence. However, it cleaves at the axis of symmetry, generating blunt ends. While XMA1 also identifies the same sequence, it is from another organism. However, it digests the sequence differently, generating sticky ends. So such restriction enzymes that can recognize the same sequence, they are from different sources. However, they cleave the sequence, generating different types of ends, are known as neoschizomers of each other. In the next slide, we will discuss about enzyme DNA ligase and also discuss about the basic cloning strategy. So here we have our plasmid that is having an origin of replication required for propagation of the plasmid. It has a tetracycline resistance marker for selection of the plasmid, and it has a multiple cloning site that can be used for cloning purposes. Similarly, we have our insert DNA molecule that can be obtained from various procedures such as digestion or genomic DNA or PCR method can be used, which are, so here we have to digest the plasmid molecule as well as the insert molecule with the same restriction enzyme so that compatible ends can be generated. We then mix the two and then allow annealing reaction to take place. And then we add the enzyme DNA ligase, the ligase buffer, and we allow the ligation reaction to take place where the mix are sealed by formation of a phosphodiester bond by the activity of enzyme DNA ligase. After formation of an intact plasmid, we perform a transformation reaction on using a suitable host cell such as E. coli. And here you can see on a Petri plate. So the Petri plate that is used over here is a medium containing antibiotic tetracycline. So only those bacteria that have picked up the plasmid are able to multiply and form colonies on the tetracycline containing medium. Two different types of DNA ligases are commonly in use, such as the E. coli DNA ligase and the T4 DNA ligase. So T4 is a bacteriophage. So some of the properties of these two enzymes are mentioned here for your reference. Also, the ligation reaction is carried out in cold conditions, generally about 4 to 8 degrees Celsius is the temperature used for a ligation reaction. So that's it about restriction enzymes and enzyme DNA ligase. Thank you very much. Kindly like, share, and subscribe if you like the content.